Hello, everyone, and welcome to today's lecture on the muscular system and types of muscles. Over the next 30 minutes, we're going to explore one of the most fundamental systems in the human body, the muscular system. Muscles are not only about physical strength or movement, though those are important aspects. They're also essential to things we don't consciously control, like the rhythm of your heartbeat, the movement of food through your digestive tract, and even the dilation of blood vessels. In other words, muscles are involved in everything from lifting weights to keeping you alive while you sleep. In today's session, we'll start with a general overview of the muscular system. Then we'll dive deeper into the three main types of muscle tissue: skeletal, cardiac, and smooth muscle. Each has its own structure, function, and unique physiological properties. Along the way, we'll also touch on how muscles work at the cellular level, how they generate force, and how they contribute to overall health, including what can go wrong when muscles are damaged or diseased. The goal isn't just to memorize types and terms, it's to understand how these tissues behave in the context of the whole human body. And by the end of the lecture, I hope you'll not only be able to distinguish between the different types of muscles, but also appreciate how they work together to make life possible. Let's get started. Before we jump into the content, let's take a moment to review the learning objectives for today's lecture. These will serve as our roadmap. By the end of this session, you should be able to understand the organization of the muscular system, from the whole body level down to the microscopic contractile units. Describe the structural and functional characteristics of the three major muscle types: skeletal, cardiac, and smooth. Identify where each muscle type is found, what they do, and how they are controlled, whether voluntarily or involuntarily. And finally, you should be able to connect these basic concepts to real-life physiology and clinical relevance that includes understanding conditions like muscular dystrophy or how a muscle contracts at the cellular level. These objectives aren't just for academic assessment. They reflect real-world knowledge that's foundational for advanced courses in physiology, medicine, kinesiology, and other health sciences. As we go through the slides, I encourage you to ask questions, make connections to prior knowledge, and think about how muscles relate to systems like the nervous system or the cardiovascular system. Interconnectivity is the key to mastering human biology. So, with our goals clearly outlined, let's begin by getting an overview of what the muscular system actually is. Let's begin by zooming out and looking at the muscular system as a whole. When we talk about the muscular system, we're referring to the collection of tissues in the body that are specialized for contraction. That contraction allows for a wide range of functions, not just movement, but also stability, circulation, digestion, and even temperature regulation. Now, most textbooks mention that there are approximately 650 named skeletal muscles in the human body, and that's not even counting all the smaller muscle groups or subdivisions. These muscles, taken together, make up nearly half of our total body mass. In fact, muscle tissue accounts for about 40% to 50% of your body weight, depending on age, sex, and physical conditioning. That's a huge investment by the body, and it tells us just how essential muscles are to our survival and function. So, what are the main functions of the muscular system? Let's go through a few key ones. Movement. This is the most obvious. Skeletal muscles pull on bones to produce voluntary movements like walking, writing, or nodding your head. But it also includes involuntary movements like reflexes, posture, and support. Even when you're sitting still, muscles are working. Postural muscles hold your body upright against gravity. Without constant low-level muscle contraction, we'd slump over like rag dolls. Joint stability. Muscles help stabilize joints by maintaining proper alignment, especially during motion. This is particularly important in joints like the shoulder, which relies heavily on muscle tone for stability. Heat generation. Muscles produce heat as a byproduct of metabolism. In fact, shivering is your body's way of generating heat via rapid, involuntary muscle contractions. This function is critical for thermoregulation. Circulation and internal movement. Not all movement is visible from the outside. Cardiac muscle pumps blood throughout the body. Smooth muscle moves food through the digestive tract, constricts blood vessels, and facilitates functions like urination or childbirth. Protection of internal organs. Muscles form protective layers around vulnerable organs. For example, your abdominal muscles shield your intestines and other visceral organs. A key point to remember is that muscles do not act alone. They work in close coordination with the skeletal system to form what we call the musculoskeletal system. They also rely heavily on input from the nervous system and support from the circulatory system for oxygen and nutrient delivery. The muscular system can thus be thought of not in isolation, but as an integrated player in whole-body physiology. Its components are highly specialized, but also highly interconnected. So now that we've laid out the why muscles are so vital, let's take a closer look at their structure, starting at the tissue level. In the next slide, we'll break down the hierarchical organization of a muscle, from the whole organ down to the microscopic contractile unit, the sarcomere. Now that we've established why muscles are so essential, let's look more closely at what a muscle is made of, from the whole muscle down to its microscopic machinery. Muscle tissue is highly organized and follows a hierarchical structure, meaning it's arranged in nested levels, each built from smaller components. Let's walk through this organization step by step. We start with the whole muscle, such as the biceps brachii. This is the organ-level structure, what you see and feel beneath the skin. Each whole muscle is wrapped in a connective tissue sheath called the epimysium. Next, within the muscle, we find bundles called fascicles. Think of these like clusters or compartments. Each fascicle is surrounded by another connective tissue layer called the perimysium. Inside each fascicle are dozens to hundreds of muscle fibers, which are individual muscle cells. Now here's something unique. Skeletal muscle cells are long, cylindrical, and multinucleated, meaning they contain multiple nuclei. These cells can stretch the entire length of the muscle. They are among the longest cells in the human body. Each muscle fiber is wrapped in a thin connective tissue layer called the endomysium. Now, going even deeper, within each muscle fiber are hundreds or even thousands of myofibrils. These are long, rod-like structures that run parallel through the cell. Myofibrils are the contractile machinery of the muscle. This is where the magic happens. Myofibrils themselves are made up of repeating units called sarcomeres, which are the basic functional and contractile units of muscle. A sarcomere is the smallest structure that can perform muscle contraction. It's composed of thick filaments, myosin, and thin filaments, actin, which slide past each other to shorten the muscle, a process we'll dive deeper into later. What's especially important here is that these sarcomeres are arranged in a very regular, repeating pattern, which gives skeletal and cardiac muscle their striated appearance under the microscope. You'll see those visible light and dark bands. That's the sarcomere structure at work. A few more key components worth mentioning. The sarcoplasm is the cytoplasm of the muscle fiber. It contains mitochondria, glycogen stores, and myoglobin, an oxygen-binding protein. The sarcolemma is the plasma membrane of the muscle cell, and it plays a vital role in conducting electrical impulses. The sarcoplasmic reticulum, SR, is a specialized form of endoplasmic reticulum that stores and releases calcium, which, as you'll see, is critical for contraction. So to summarize, a muscle is made up of fascicles, which contain muscle fibers, which house myofibrils, which are composed of sarcomeres. It's a beautiful example of biological organization, from macroscopic strength all the way to microscopic precision. Now that we've covered the structural organization of muscle, let's shift gears and talk about the different types of muscle tissue found in the human body. There are three main types: skeletal, cardiac, and smooth muscle. Each has distinct structural, functional, and regulatory characteristics, and they're found in different parts of a body serving unique roles. Let's start with a high-level comparison, and I'll walk you through the key differences column by column in this table. 1. Striation pattern. Skeletal and cardiac muscles are striated, meaning they have visible bands under the microscope. These stripes come from the organized arrangement of sarcomeres, those repeating units of actin and myosin we discussed earlier. In contrast, smooth muscle is non-striated. Its contractile proteins are still present, but they're arranged in a less organized, more crisscrossed fashion, which gives the tissue a smooth appearance under the microscope. 2. Control. Voluntary versus involuntary. Skeletal muscle is under voluntary control. That means you consciously decide when to move your arm or walk. Signals from your somatic nervous system initiate contraction. Cardiac and smooth muscles, on the other hand, are both involuntary. You don't have to think about your heart beating or your intestines digesting food. These are controlled by the autonomic nervous system and internal pacemakers. 3. Nuclei. Skeletal muscle fibers are multinucleated, a result of multiple myoblasts, embryonic muscle cells, fusing during development. These nuclei are located at the periphery of the cell. In contrast, cardiac and smooth muscle cells each typically have a single nucleus, and it's usually centrally located. 4. Shape and arrangement. Skeletal muscle fibers are long, cylindrical, and often unbranched. Cardiac muscle cells are branched and interconnected, forming a syncytium through intercalated discs, specialized junctions that allow for coordinated contraction. Smooth muscle cells are spindle-shaped, thick in the middle and tapering at the ends, and they often form layers in the walls of hollow organs like blood vessels and the digestive tract. 5. Location and function. Skeletal muscle is attached to bones and is responsible for movement, posture, and heat generation. Cardiac muscle is found only in the heart, where it functions to pump blood continuously. Smooth muscle lines the walls of internal organs and blood vessels, regulating things like blood flow, digestion, and airway diameter. Now, this table gives us a great side-by-side framework, but to really understand these tissues, we'll need to explore each in depth: how they look under the microscope, how they function, and how they're adapted to their unique roles. Let's now take a closer look at skeletal muscle, the type we typically associate with movement and strength. Skeletal muscle is the most abundant muscle type in the body, and it's what gives the body its form and mobility. This is the muscle tissue that attaches to bones via tendons and allows us to carry out voluntary movements, things like walking, lifting, typing, or even smiling. It's also responsible for maintaining posture and generating heat through metabolic activity. Structurally, skeletal muscle is composed of long, cylindrical fibers that run the entire length of a muscle. These fibers are incredibly specialized. Unlike most cells in the body, skeletal muscle fibers are multinucleated. Why is that? It's because they form from the fusion of many smaller embryonic cells called myoblasts, and each contributes its nucleus to the final cell. These nuclei are pushed out to the periphery of the cell, just beneath the sarcolemma, the muscle cell membrane. Under the microscope, skeletal muscle displays a distinct, striated appearance. This is due to the highly ordered arrangement of sarcomeres, the repeating units within myofibrils that contain actin and myosin filaments. These filaments give the muscle its ability to contract in a very organized and powerful way. The alternating light, I bands, and dark, A bands, stripes you see are the physical representation of these overlapping filaments. Each muscle fiber is encased in endomysium, and groups of fibers form fascicles, surrounded by perimysium. Finally, the entire muscle is wrapped in epimysium. This connective tissue network not only organizes the muscle but also allows nerves and blood vessels to penetrate the muscle tissue, ensuring each fiber receives oxygen, nutrients, and neural input. Skeletal muscle also has a rich blood supply and is closely connected with the somatic nervous system, which allows for precise and coordinated control. Every muscle fiber is connected to a motor neuron via a structure called the neuromuscular junction, which we'll look at in more detail when we discuss contraction. Functionally, skeletal muscles work by pulling, not pushing. They operate on a lever system, with bones acting as levers and joints as fulcrums. For movement to occur, muscles must work in pairs or groups. The agonist performs the main action. The antagonist opposes that action. Synergists assist the agonist, and fixators stabilize the joint. Let's take a classic example. The biceps brachii flexes the elbow, while the triceps brachii extends it. These two muscles act in opposition to control forearm movement. Another important aspect is adaptability. Skeletal muscle can hypertrophy, grow larger and stronger with resistance training. On the other hand, it can atrophy if it's used for long periods, such as during immobilization or bed rest. So to summarize, skeletal muscle is a voluntary, striated, multinucleated tissue adapted for rapid, forceful contraction. It's highly organized, tightly controlled by the nervous system, and capable of remarkable plasticity in response to physical demands. Now that we've examined the structure of skeletal muscle, it's time to understand how it works, how it converts electrical signals into mechanical force. In other words, how do these cells actually contract? The process of skeletal muscle contraction is known as the excitation-contraction coupling mechanism. It involves several finely coordinated steps, starting with a signal from the nervous system and ending with sarcomere shortening. Let's break it down step by step. 1. Neural stimulation. The role of the neuromuscular junction (NMJ). Every skeletal muscle fiber is innervated by a motor neuron. The connection point between the neuron and the muscle fiber is called the neuromuscular junction. When a motor neuron fires an action potential, it travels down the axon to the axon terminal. There, it causes the release of a neurotransmitter, acetylcholine, ACH, into the synaptic cleft. ACH binds to receptors on the sarcolemma, specifically at the motor end plate of the muscle fiber, causing sodium, Na, channels to open and depolarizing the membrane. This initiates an action potential in the muscle cell, which then spreads across the sarcolemma and down into the T-tubules, deep invaginations of the membrane that penetrate into the interior of the muscle cell. 2. Calcium release. The role of the sarcoplasmic reticulum. This action potential triggers the sarcoplasmic reticulum, SR, a specialized organelle that stores calcium ions, to release Ca superscript 2 into the sarcoplasm. Calcium is the key signal for contraction. When it floods into the muscle cell, it binds to a regulatory protein called troponin, which is attached to another protein called tropomyosin that normally blocks the binding sites on actin. When troponin binds calcium, it changes shape, causing tropomyosin to shift and expose the myosin-binding sites on the actin filament. 3. Cross-bridge cycling, the power stroke. Now that binding sites are exposed, myosin heads, which are already energized by ADP, can attach to actin, forming what's known as a cross-bridge. The myosin head then pivots, pulling the actin filament inward. This movement is called the power stroke. After the stroke, a new ADP molecule binds to the myosin head, causing it to detach from actin. The ADP is then hydrolyzed, recocking the head into position for another cycle. This process of binding, pivoting, detaching, and resetting continues as long as calcium and ADP are present, and it results in the shortening of the sarcomeres and ultimately the entire muscle fiber. 4. Relaxation. Once the neural signal stops, acetylcholine is broken down by an enzyme called acetylcholinesterase, and calcium is pumped back into the sarcoplasmic reticulum using active transport. With calcium gone, troponin and tropomyosin return to their original conformation, covering the binding sites on actin and effectively ending contraction. Energy requirements. Muscle contraction requires large amounts of ADP, not just for the power stroke, but also for detaching myosin, pumping calcium back into the SR, and restoring ionic balances after depolarization. Muscle fibers use several systems to generate ADP. Creatine phosphate provides immediate energy. Anaerobic glycolysis offers short bursts. Aerobic respiration is the most sustainable long-term source. To summarize, skeletal muscle contraction is a beautifully orchestrated process involving the nervous system, electrical signals, calcium ions, ADP, and the structural proteins inside the sarcomere. It's fast, efficient, and precisely controlled, which is exactly what we need for voluntary movement. Now, we move from the voluntary skeletal muscle to the cardiac muscle, a tissue specialized for the continuous, rhythmic contractions that keep your heart beating, pumping blood throughout your entire body, every second of your life. Cardiac muscle shares some features with skeletal muscle, but it also has unique structural and functional adaptations that make it perfectly suited to its role. Structurally, cardiac muscle fibers are striated, like skeletal muscle, meaning they have the characteristic banding pattern due to the organized sarcomeres. However, cardiac muscle cells are shorter and branched, unlike the long, cylindrical skeletal muscle fibers. This branching creates a highly interconnected network of cells, linked together by specialized junctions called intercalated discs. These discs are critical for both mechanical and electrical coupling between cells. Intercalated discs contain desmosomes, which physically hold adjacent cells together, ensuring the heart tissue can withstand the continuous mechanical stress of beating. Gap junctions, which form channels that allow ions to pass directly from one cell to the next. This electrical connectivity enables the rapid and coordinated spread of action potentials across the heart muscle, ensuring that the heart contracts as a unified, synchronized pump rather than a collection of isolated cells. Cardiac muscle cells typically have one central nucleus, although occasionally two nuclei can be present. Unlike skeletal muscle, cardiac muscle fibers are not multinucleated. Because the heart must work continuously without fatigue, cardiac muscle fibers have an abundance of mitochondria, far more than skeletal muscle, to meet their high-energy demand through aerobic respiration. Functionally, cardiac muscle is involuntary and autorhythmic. This means it contracts without conscious effort and can generate its own electrical impulses. The heart's pacemaker cells in the sinoatrial SA node set the rhythm of contraction, which is then spread throughout the myocardium via the conduction system and intercalated discs. Although cardiac muscle contractions are similar to skeletal muscle at the cellular level, involving sliding filaments and calcium-dependent processes, the timing and regulation differ. The heart's contraction cycle includes a long refractory period, which prevents tetanus, a sustained contraction that would be fatal if it happened in the heart. In summary, cardiac muscle is uniquely designed for endurance, reliability, and rapid coordinated contraction. It is striated but branched. It has intercalated discs for electrical and mechanical connectivity. It operates involuntarily, with intrinsic rhythmicity. It relies almost entirely on aerobic metabolism to sustain its energy needs. This design ensures your heart beats rhythmically and forcefully without fatigue, tirelessly pumping blood to nourish every cell in your body. Let's focus on how the cardiac muscle functions to keep the heart beating reliably and efficiently, based on a few critical physiological principles. 1. SA node and electrical impulse generation. At the heart of cardiac muscle physiology is the sinoatrial SA node, often called the heart's natural pacemaker. This specialized cluster of cells in the right atrium spontaneously generates electrical impulses without any external stimuli. These impulses initiate the heartbeat by triggering the contraction of the atrial muscle fibers. From the SA node, the electrical signal rapidly spreads through the atria, causing them to contract and push blood into the ventricles. The impulse then travels through the atrioventricular AV node and down the conduction system to stimulate ventricular contraction. This self-generated rhythmic impulse is fundamental. It ensures the heart beats continuously and autonomously, adapting rate based on the body's demands without conscious effort. 2. Long refractory period prevents tetanus. An essential feature that distinguishes cardiac muscle from skeletal muscle is the long refractory period. After a contraction, cardiac muscle cells require a relatively extended time before they can be stimulated again. This refractory period is vital because it prevents tetanus, which is a sustained contraction seen in skeletal muscles during rapid stimulation. If tetanus were to occur in the heart, it would lose its ability to relax between beats and pump blood effectively, which would be life-threatening. The long refractory period thus ensures that each contraction is followed by relaxation, allowing the heart chambers to fill properly before the next contraction. 3. Role of calcium from sarcoplasmic reticulum and extracellular fluid. Cardiac muscle contraction depends heavily on calcium ions, but in a unique way compared to skeletal muscle. When an action potential travels across the cardiac muscle cell membrane, it opens voltage-gated calcium channels, allowing calcium to enter from the extracellular fluid into the cytoplasm. This influx of calcium then triggers the release of more calcium from the sarcoplasmic reticulum inside the cell, a process called calcium-induced calcium release. The sudden rise in intracellular calcium binds to troponin, causing conformational changes that expose myosin-binding sites on actin, enabling the contractile machinery to work. Because the contraction depends both on extracellular calcium entry and internal calcium release, cardiac muscle contraction can be finely regulated and sustained with a rhythmic and forceful beat. In summary, the SA node acts as the heart's rhythm generator. The long refractory period protects the heart from tetanic contractions, and the dual source of calcium ions from both extracellular fluid and the sarcoplasmic reticulum ensures precise and efficient contraction. Together, These features enable the heart to beat reliably over a lifetime, adapting to physiological demands without fatigue. Let's now explore the third major type of muscle tissue, smooth muscle. Unlike skeletal and cardiac muscles, smooth muscle is specialized for slow, sustained, involuntary contractions in many internal organs. Structure. Smooth muscle cells are quite distinct in appearance and organization compared to the other muscle types. These cells are spindle-shaped, meaning they are elongated with tapered ends, and typically have a single, centrally located nucleus. Unlike skeletal and cardiac muscle, smooth muscle is non-striated. This means that it lacks the regular banding pattern seen in the other muscles because its actin and myosin filaments are not arranged into sarcomeres. Instead, these contractile proteins are arranged in a more irregular, lattice-like network throughout the cytoplasm. This arrangement allows smooth muscle to contract in multiple directions, which is especially useful for the diverse roles it plays in different organs. Smooth muscle cells are often organized into sheets or layers, forming the walls of hollow organs such as blood vessels, the gastrointestinal tract, the bladder, and the uterus. The cells are connected by gap junctions, enabling coordinated contraction across the tissue. Function. Smooth muscle contraction is involuntary, controlled by the autonomic nervous system and influenced by local chemical signals, hormones, and stretch. Its main roles include regulating the diameter of blood vessels, moving food through the digestive system via peristalsis, controlling airflow in the respiratory tract, and regulating bladder emptying, among others. Because smooth muscle contractions are generally slower and more sustained than those of skeletal or cardiac muscle, it is well suited for maintaining tonic contractions or rhythmic contractions over long periods without fatigue. Contraction mechanism. Though smooth muscle contraction also depends on the interaction of actin and myosin filaments, the process differs from skeletal muscle. Smooth muscle uses a regulatory protein called calmodulin rather than troponin, and contraction is initiated when calcium binds to calmodulin, activating myosin light chain kinase. This enzyme phosphorylates myosin heads, enabling them to bind to actin and generate contraction. Because of this, smooth muscle can sustain contractions with less energy consumption, which is essential for its function in organs that require continuous tone or slow, controlled contractions. Summary. Spindle-shaped, single-nucleus cells. Non-striated, with irregular filament arrangement. Found in walls of hollow organs. Involuntary control via autonomic nervous system and local signals. Slow, sustained contractions ideal for regulating organ function. Smooth muscle's unique structure and functional properties allow it to perform vital roles that skeletal and cardiac muscles cannot, keeping essential internal processes running smoothly. Let's now take a closer look at the physiology of smooth muscle. Unlike skeletal and cardiac muscle, smooth muscle operates entirely involuntarily. It's controlled by the autonomic nervous system, but also responds to hormones, local chemical signals, and mechanical stretch. This gives it remarkable flexibility across different organs. Contraction begins when calcium ions, Ca2, enter the smooth muscle cell from either the extracellular space or the sarcoplasmic reticulum. But unlike skeletal muscle, there's no troponin. Instead, calcium binds to calmodulin, which activates myosin light chain kinase, MLCK. This enzyme phosphorylates myosin, allowing it to bind actin and produce contraction. Smooth muscle contractions are generally slower and more sustained than in skeletal or cardiac muscle. There are two main patterns. Phasic contractions, which are rhythmic and wave-like, like in the intestines during peristalsis. Tonic contractions, which are long-lasting, such as in blood vessels to maintain blood pressure. Another key feature is plasticity. Smooth muscle can stretch significantly and still maintain tension. For example, the bladder expands as it fills but only contracts when it's time to empty. In some tissues, smooth muscle exhibits slow-wave potentials, spontaneous electrical rhythms that help coordinate contractions, often regulated by pacemaker-like cells. So overall, smooth muscle is built for durable, low-energy contractions, perfectly suited for regulating the function of internal organs over long periods without fatigue. Let's take a closer look at how muscles coordinate during a simple but familiar action, elbow flexion, like when you bring your hand toward your shoulder. This movement is a great example to illustrate the 4 key roles that muscles can play: agonist, antagonist, synergist, and fixator. Let's start with the agonist, or prime mover, the main muscle responsible for the movement. In elbow flexion, the biceps brachii takes this role. When you lift something or bend your elbow, the biceps contracts, pulling the forearm up toward the shoulder. It generates the majority of the force needed for that motion. Now for the antagonist, this is the muscle that opposes the action of the agonist. In this case, it's the triceps brachii. While the biceps contracts to flex the elbow, the triceps relaxes, allowing the movement to occur. However, it doesn't go completely limp. It may maintain a small amount of tension to ensure the motion is controlled and smooth. When you reverse the movement, extending the elbow, the roles switch. Triceps becomes the agonist, and biceps becomes the antagonist. Next, we have synergist muscles. These assist the agonist by adding force or helping guide the movement. In elbow flexion, muscles like the brachialis and brachioradialis act as synergists. They don't produce the primary movement but support and refine it, either by increasing the total force or helping the movement track properly. Lastly, the fixator. Fixators don't move the joint in question, but instead stabilize other nearby joints or bones so the agonist can work efficiently. During elbow flexion, muscles like the rotator cuff group, particularly the infraspinatus and supraspinatus, act as fixators. They stabilize the shoulder joint, anchoring the origin of the biceps so that it can contract effectively without causing unnecessary shoulder movement. So in this action of elbow flexion, the muscle roles break down like this. Agonist, biceps brachii, main mover for flexion. Antagonist, triceps brachii, relaxes to allow flexion. Synergists, brachialis and brachioradialis, assist with movement. Fixators, rotator cuff muscles, stabilize the shoulder joint. This functional teamwork ensures your movements are precise, efficient, and safe. It's this coordination that lets you lift a cup of coffee smoothly or catch a ball without injuring yourself. As we study movement, understanding these roles is fundamental not just in anatomy, but in areas like physical therapy, sports science, and clinical medicine. Let's now talk about something that's often overlooked in muscle discussions, but absolutely vital for daily function: muscle tone and posture. First, what is muscle tone? In physiological terms, muscle tone refers to the slight, continuous contraction of muscles, even when we're not consciously moving. This isn't the same as voluntary contraction like lifting a weight. Instead, muscle tone is a baseline level of tension maintained by the nervous system to keep muscles ready for action. It's what gives muscles their firmness and allows us to respond quickly when needed. Even when you're sitting perfectly still in class, your muscles are at work. The muscles in your back, neck, abdomen, and legs are subtly contracting to hold you upright. This is due to reflex activity involving spinal reflex arcs and input from the muscle spindles, sensory receptors that detect changes in muscle length and send signals to adjust tone accordingly. This helps your body maintain equilibrium and resist the constant pull of gravity. Now, let's connect this to posture. Good posture is the result of balanced muscle tone across muscle groups. For example, in standing posture, back extensors and abdominal muscles work together to keep the spine erect. Hip flexors and extensors balance the pelvis. Muscles of the neck and shoulders keep the head aligned over the torso. When muscle tone is too low, a condition called hypotonia, the body appears floppy and struggles to support posture. This can occur in various neuromuscular conditions. On the other hand, hypertonia, excessive tone, can lead to stiffness, restricted movement, and postural abnormalities. This is common in conditions like spastic cerebral palsy or after a stroke. Importantly, muscle tone is regulated largely by the central nervous system, particularly the brainstem and spinal cord. Disorders affecting these areas often present with changes in tone and postural instability. Muscle tone also plays a role in energy efficiency. By maintaining posture automatically, your body conserves energy that would otherwise be spent on full contractions. Think of it as a muscle idle mode, ready to engage fully but operating efficiently in the background. Lastly, posture isn't static, it constantly adjusts. Standing on one leg, balancing on uneven terrain, or even sitting upright for long periods all require dynamic postural control, which is finely tuned by sensory input from vision, proprioception, and the vestibular system. So, to sum up, muscle tone is an ongoing low-level contraction regulated by reflexes and the nervous system. It enables posture, providing stability and readiness for movement. Healthy tone and posture are essential for balance, mobility, and injury prevention. Understanding this helps us see that muscles don't just move us, they also hold us together, quite literally. Now let's talk about something many students and athletes are curious about: how muscles grow and repair. Muscle is a dynamic tissue, constantly remodeling in response to stress, injury, hormones, and activity. There are two major terms we use when describing muscle growth: hypertrophy and hyperplasia. Small blue diamond hypertrophy is the increase in the size of existing muscle fibers. This is the primary way skeletal muscle grows in humans. When you lift weights or perform resistance training, you create tiny microtears in your muscle fibers. In response, the body repairs them, and while doing so, it reinforces the fibers, making them thicker and stronger. This increased cross-sectional area results in visible muscle growth. Small blue diamond hyperplasia, on the other hand, refers to an increase in the number of muscle fibers. There's limited evidence that this occurs to a significant extent in adult human muscle. Most of our gains come from hypertrophy. That said, some animal studies suggest hyperplasia can occur under extreme loading or certain hormonal conditions. Now, let's introduce the satellite cells, the real stars of muscle repair. These are quiescent stem-like cells located between the sarcolemma and basal lamina of muscle fibers. When muscle is damaged from exercise or injury, satellite cells become activated. They proliferate, differentiate into myoblasts, and fuse either with existing fibers, promoting hypertrophy, or with each other to form new fibers in severe damage. They're essential not just for repair, but also for adaptive muscle growth. The degree of muscle growth and repair is strongly influenced by both exercise and hormones. Resistance training stimulates hypertrophy by inducing mechanical tension, metabolic stress, and microtrauma. Endurance training doesn't significantly increase fiber size, but enhances mitochondrial density and capillarization. Hormones like testosterone, growth hormone, and insulin like growth factor 1, IGF-1 play major roles by activating anabolic signaling pathways that enhance protein synthesis and satellite cell activity. Also important is the balance between synthesis and degradation. After exercise, protein synthesis must outweigh breakdown for net muscle gain to occur. This is why nutrition, rest, and recovery are crucial parts of the muscle-building process. As we wrap up the main content of this lecture, it's important to acknowledge what happens when the muscular system malfunctions. Today we'll briefly cover several key muscular disorders, from genetic conditions to acute injuries, to show how complex and vulnerable this system can be. 1. Muscular dystrophy, MD. Muscular dystrophy refers to a group of inherited disorders characterized by progressive degeneration and weakness of skeletal muscles. The most common type is Duchenne muscular dystrophy, DMD, caused by mutations in the dystrophin gene. Dystrophin is a structural protein that helps stabilize muscle fibers during contraction. Without it, muscle cells become damaged and eventually die. DMD primarily affects boys and becomes evident in early childhood, with progressive loss of mobility. There's no cure yet, but gene therapy, physical therapy, and steroids can help slow progression. 2. Myasthenia gravis, MG. This is an autoimmune disorder where the body produces antibodies against acetylcholine receptors at the neuromuscular junction. This disrupts communication between nerves and muscles, leading to muscle weakness, especially in the face, eyes, throat, and limbs. A hallmark symptom is muscle fatigue that worsens with activity. MG can be managed with immunosuppressants, anticholinesterase drugs, and sometimes thymectomy, depending on the underlying cause. 3. Muscle cramps and strains. These are very common, often benign but can be painful and functionally limiting. Cramps are involuntary, sustained muscle contractions, often caused by dehydration, electrolyte imbalances, overuse, or poor circulation. They usually resolve on their own, but can be relieved with gentle stretching or hydration. Strains are injuries to muscle fibers or tendons, often resulting from overstretching or excessive load. They vary in severity from mild, grade I, to complete tears, grade 3. Treatment includes RICE, rest, ice, compression, elevation, followed by gradual rehabilitation. 4. Rhabdomyolysis, brief overview. Rhabdomyolysis is a serious and potentially life-threatening condition where skeletal muscle breaks down rapidly, releasing myoglobin into the bloodstream. This can damage the kidneys and cause complications like electrolyte imbalance or renal failure. Causes include crush injuries, extreme exertion, certain drugs, or heat stroke. Early symptoms include muscle pain, weakness, and dark urine. It's a medical emergency requiring aggressive fluid therapy. These disorders range from genetic and autoimmune conditions to acute injuries and metabolic breakdowns, but they all illustrate the importance of maintaining muscular health and recognizing early warning signs. So, whether it's a rare genetic disease like muscular dystrophy or a common muscle strain from poor lifting technique, each example reminds us how essential proper muscle function is and how dependent it is on neurological input, structural integrity, and metabolic balance. In this slide, we'll walk through the most commonly used clinical and laboratory methods for assessing muscle structure and function. 1. Electromyography, EMG. This is one of the most direct tools for assessing muscle function. EMG involves inserting a fine needle electrode into the muscle to record its electrical activity at rest and during contraction. In healthy muscle, you see minimal activity at rest and a characteristic pattern during contraction. In conditions like myasthenia gravis or neuropathies, EMG reveals abnormal or diminished activity. EMG is especially useful in distinguishing between nerve versus muscle pathologies. 2. Serum creatine kinase (CK) levels. Creatine kinase is an enzyme released from damaged muscle cells. Elevated CK levels in the blood are a red flag for muscle injury, inflammation, or degeneration. In muscular dystrophy, CK levels may be markedly elevated, even before symptoms are noticeable. CK is also elevated in rhabdomyolysis, making it a vital early biomarker in emergency settings. 3. Muscle biopsy. In some cases, a small sample of muscle tissue is taken surgically and examined under a microscope. This can provide direct evidence of structural changes, such as fiber degeneration or regeneration, inflammatory cell infiltration, and abnormal protein accumulation. Muscle biopsies are particularly useful for diagnosing muscular dystrophies, myositis, and certain mitochondrial disorders. They can also be used for genetic analysis, especially when inherited muscle disease is suspected. Alright, so we've covered a lot about how muscles work, from their structure to how they contract, grow, and even how they sometimes break down. But let's step back for a moment and ask, why does all this matter in real life? First, let's talk health. Your muscles aren't just for movement. They're critical to your overall wellness. For example, maintaining good muscle tone and strength can help prevent falls in older adults, improve posture, and support joint health. In physical therapy, we use what we know about muscle function to rehabilitate injuries, restore mobility, and even help people recover from strokes or surgeries. Even for people with chronic diseases like type 2 diabetes, regular muscle use helps regulate blood sugar levels, because skeletal muscle is one of the biggest consumers of glucose in the body. So just moving your muscles regularly becomes a form of preventive medicine. Now, let's shift to performance. Whether someone's an elite athlete or just wants to lift a bit more at the gym, understanding muscle physiology helps them train smarter. For example, want more strength? Focus on hypertrophy and resistance training. Want better endurance? Target slow-twitch fibers with aerobic exercise. Need faster recovery? Know how inflammation and repair cycles work. Even technologies like electromyography, which we talked about earlier, are now used in sports science to measure muscle activation during training and improve biomechanics. And then there are more unique settings like space travel. In microgravity, astronauts lose muscle mass quickly. So researchers use their knowledge of muscle biology to develop resistance-based workouts for space missions. The bottom line? Whether it's improving health, enhancing athletic performance, or even designing physical rehab plans, understanding muscles is not just academic, it's practical and deeply connected to real-world outcomes. Okay, let's wrap things up with a quick recap. We started by looking at the three main types of muscles: skeletal, cardiac, and smooth, and how each has a unique role. Skeletal muscle moves our bodies, cardiac keeps the heart beating, and smooth muscle handles things like digestion and blood flow. Then we explored how muscles contract, focusing on actin and myosin, the role of calcium, and how ADP powers it all. We also talked about muscle fiber types, motor units, and what makes cardiac and smooth muscle special. Next, we got into how muscles actually make movement happen through agonists, antagonists, and helpers like synergists and fixators. We even used a simple example, your biceps and triceps, to bring it to life. We touched on how muscles maintain tone and posture, and how they grow and repair through hypertrophy and satellite cells, and how exercise and hormones play a big role in that. Then we looked at common muscle disorders like muscular dystrophy, myasthenia gravis, and muscle cramps, and the tools we use to diagnose them, like EMG, blood tests, and muscle biopsies. Finally, we talked about why this all matters for health, for performance, and for understanding how our bodies stay strong and functional. So whether you're thinking clinical, athletic, or just curious, understanding muscle is key to understanding movement and health. Thank you for listening. The comment section is open for questions. Also, don't forget to subscribe for more videos.